In this video, we will continue our discussion about the role of E-cadherins towards Invadopodia formations. So here, in today's video, we will discuss the functionality of E-cadherin, more precisely its adhesiveness that is essential for Invadopodia formations. In order to check its functional property that cause these Invadopodia formations, what it is, they prepared a BXPC3 cell lines in which the E-cadherin has been knocked down using SH E-cadherin RNAs. And this in the panel A they shows the results where they show that E-cadherins are successfully been down regulated compared to the control. And because of this down regulation of the E-cadherins, they are able to see the degradation areas per cells also goes down, and the number of Invadopodia formations per cells also goes down. Both of these things indicates that absence of the E-cadherins is causing decrease in the Invadopodia, and these things are in line with the results that we have discussed in our previous video. After that, from the literatures, they are able to see that penalty is not about that thing. So in the penalty, what they did is they overexpressed the E-cadherins in these stable E-cadherins knockdown pancreatic cancer cells. Here they in this experiment, for these experiments they have used DXPC3 pancreatic cancer cell lines. So in the compared to the mock where E-cadherin has been knocked down, when they overexpress the E-cadherins, they are able to see the increase in the degradations. It can be seen from the figure in the panel D, and also the quantifications of this shown in the panel E, where we can see that the degradations area per cells increases as soon as this E-cadherin is been overexpressed. And after that they check, once validating that E-cadherin has a direct relationship, now they wanted to check its functionality, what function of the E-cadherin that is causing the Invadopodia formations. So they check the literatures and they are able to find out that E-cadherin interactions with beta-catenin is important for its function. So what they did is they did the immunofluorescence, and they are able to see that as E-cadherin and beta-catenin are co-expressed, and these things they have also quantified using the gray value, where we can see that the presence of PLA, E-cadherins and beta-catenin. So once validating that E-cadherins is interacting with the beta-catenin, now from the literatures they are able to find out that there are two artificial inhibitors for that prevent the interactions of E-cadherins with this beta-catenin. It is AS9 and AS11. And when they use these inhibitors, they are able to see a decrease in the positive cells for Invadopodia for total cells compared to the control case where they use DMSO. And these results are true for both the inhibitors, that is AS9 and AS11. Now they checked the percentage rate of effect on the Invadopodia cells, and they are able to see that this number of Invadopodia rate of Invadopodia or number of Invadopodia per cells decreases as soon as they use the inhibitors that prevents the interactions of E-cadherins and beta-catenin. We can see that the slope of the AS11 treated cell lines has less slopes for the Invadopodia formations compared to the control case, that is the AS11 inhibitor cell lines has a slope of 0.325, whereas the DMSO or the control treated cell lines has a slope of 1.346. Once they remove the these inhibitors, and after 8 hours they are able to see that the rate of Invadopodia per cells catches up with the rate of Invadopodia per cells for this previously inhibitor treated cell lines, that is AS11, catches up with the rate of Invadopodia per cells for the control case. As we can see, the slope for the control is around, after 8 hours of latency. They can see the slope of the control is around 0.509, and slope of this Invadopodia, previously this E-cadherin's inhibitor treated cells is around 0.567. This indicates that the effect of these inhibitors are irreversible, and the interactions of E-cadherin and beta-catenin plays a role towards these Invadopodia formations. So I hope you understood how they validate the functionality of the E-cadherins, or E-cadherin adhesiveness plays an important role towards Invidopodia formations. So if you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.